Hello everyone. So we look at another session on membrane structure with the focus being on cholesterol as one of the components of the membrane lipids. So let us look at how sterols play an important function or important role in the functioning of the membrane per se. So let us look at the learning outcomes of the session. Sterols are important components of the membrane. Cholesterol is mainly present in the animal cells. You will not find it in plant cells and bacteria, but you will find other sterols present in plants. Say, for example, stigma sterols is present in plants, and bacteria, say, have hopanoids that are present that are different, much different from what cholesterol is in animal cells, but they are sterols that are contributing to the function of the membranes, whether it is bacteria, whether it is plants, whether it is animal cells. Of course, in this session, the focus is mainly going to be on cholesterol um, because we are looking at the membrane per se with respect to mainly the animal cells. Now, here and there, we will definitely look at sterols and other systems. Sterols have a relatively different structure than phospholipids and sphingolipids, yet they have a hydroxyl group and a hydrophobic component to make them amphipathic. So this is important because almost no, all lipids present in the bilipid layer are amphipathic in nature. Cholesterol has been reported to modulate the bilayer structure in multiple ways, such as changes in fluidity, thickness, compressibility, water penetration, membrane curvature, etc. Cholesterol is able to induce phase separation of the membranes in response to different conditions and interactions. So let us look at the basic structure of cholesterol first. So when you look at the structure of cholesterol, you will find that it has a small hydrophilic head and you have a steroid ring with a hydrocarbon chain that is forming the entire hydrophobic tail. So this makes it amphipathic because here is where you have the hydrophilicity and here is where you have the hydrophobicity. So this part can interact with the bilipid layer, whereas this can be part of the bilipid layer, but either facing the extracellular side or facing the inside of the cell, which are both aqueous environments per se. So you can have cholesterol present both in the outer leaflet of the membrane as well as in the inner leaflet of the membrane. Now, just looking at the ring structure, this is what the typical ring structure is. And this is how you have the OH group present over here, which is forming the polar group. And here you have a short a hydrocarbon chain that is together with the ring forming the hydrophobic tail. Now, interestingly, this ring structure is planar and it is rigid. So it does not really have too much of flexibility, but this hydrocarbon chain is flexible, and therefore this can interact with all the flexible acyl chains of phospholipids, etc. Now, these hydrophobic region is able to interact preferentially with phospholipids having saturated fatty acids, also sphingolipids having saturated acyl groups, and they can also interact with proteins. So therefore, cholesterol can interact with all the other components of the bilipid layer, and that makes it extremely important because that contributes to the dynamism of the membrane. Now, when you look at cholesterol being present in the membrane, suppose you consider a membrane which is devoid the cholesterol, then what one can observe is that there is a lot of space that is there between the phospholipids. That is thanks to one of the fatty acid chains having a cis double bond, because of which you have a kink, and that kink enables spacing out the phospholipids present in the membrane. So when you have the cholesterol being added, now the cholesterol, this ring per se is flat and rigid, and therefore what it does is it kind of reduces the mobility of the neighboring fatty acyl chains that are present. Thereby, what happens is this rigid ring structure is considerably going to reduce the movement of the neighboring lipid acyl chains, and also their ordering them. It's a kind of defined order. So they're more ordered rather than random, as you can see over here. And therefore, what happens is it will reduce the fluidity of the membrane. So you can kind of say that it is contributing to the rigidity of the membrane. You can see how the space, okay, that is there between every lipid moiety is going to be less, because now you have the cholesterol here in between, and this is the other lipid. So you can see that there is hardly any space between these two. So when you have, equal number of cholesterol, or you have a ratio of 1:1 with respect to cholesterol and phospholipids, you will find that the membrane is quite rigid. So therefore, sterols can be a means of increasing the tolerance of the membrane to a particular environment or to a particular stress. Basically, because the membrane becomes more rigid, less permeable, and hence it protects the inner environment of the cell more when it has a lot of cholesterol present. But of course, it doesn't necessarily mean that you should always have one cholesterol unit per phospholipid unit. That can vary from cell to cell, that can vary from membrane to membrane as well. And within the membrane itself, you can have certain regions of the membrane having more amount of cholesterol, and certain places in the membrane having no cholesterol or very little cholesterol as well. Again, looking at this factor, that when you have the cholesterol inserted within the and interacting with the phospholipids, what happens is, because it becomes a very highly ordered structure, okay, without cholesterol and phospholipid interaction. So because there is no cholesterol, there is no cholesterol phospholipid interaction. This is considered to be a liquid disordered phase. So we're calling it liquid because we know that the membrane is fluid, and therefore we're calling this a very highly disordered phase. There is no order, okay, in and there's very randomization that is there, and so therefore this is called as a liquid disordered phase. But the moment you add cholesterol, this becomes highly ordered, okay, and therefore this, where you have cholesterol and phospholipid interaction, this portion of the membrane becomes, or is considered to be, a liquid ordered phase. So this is cholesterol induced liquid ordered phase. This, because there is no cholesterol, you have no ordered phase per se. And because of this order being created, what is also observed is that the diameter, or this distance, sorry, not distance, not diameter, the distance that is there over here is more than the distance over here. And so you will find that the thickness of the membrane has also increased because of the presence of the cholesterol. So this is an important fact that is to be noted. One, that it influences the dynamics of the membrane. It changes the fluidity of the membrane. It can make it either more fluid or more rigid depending on how it is present in the membrane, and it can increase the thickness of the membrane as well. Thereby, it is also affecting the bending and the compressibility of the membrane. So when you look at the liquid disordered phase, compare it with the liquid ordered phase, you will find that the compressibility of this membrane is much more than the compressibility of this membrane. So the lipid ordered phase, which is due to the cholesterol, makes the membrane less compressible. It becomes more rigid. So you become you are able to compress it lesser. So therefore, that is one thing that the cholesterol does. And it also increases the viscosity, because you have more the lipid components. You know that more solute content in solvent makes the entire thing more viscous. So the viscosity is also going to increase, and that is going to decrease, the lateral diffusion of the phospholipids, lateral diffusion of the surrounding lipids. It's also going to affect the rotational diffusion of the surrounding lipids. So movement is also curtailed, movement is also restricted because of the presence of the cholesterol. Interestingly enough, when you have no cholesterol, you will find that the membrane is more permeable to water, while if you have cholesterol, it is less permeable. So the penetration, water penetration, is much lesser when a membrane has cholesterol present. Again, another important fact is that, we know that lipids have an intrinsic shape of their own. So if you consider a membrane with just phospholipids, say for example, and that too if it is phosphatidylcholine, or if it is sphingomyelin, then you would observe that they have cylindrical structure, so it has a flat kind of structure. Now when you put within them cholesterol having a very small hydrophilic head, it is only that OH group over there. Because of that, it adopts what is called as a conical structure. So the presence of conical structure therefore will lead to a certain kind of curvature in the membrane. So in fact, what has been observed, regions where the cell membrane has to kind of invaginate to endocytose food, or to endocytose a foreign particulate matter, or to endocytose even liquid droplets, it has to curve, So in these curved regions, the portion where it is curved, you will find that cholesterol is contributing to spontaneous curvature of the membrane. So it does help in the curvature of the bilipid layer. Now again over here, when you see that you have membranes which is made up of phospholipids and sphingolipids, and if you have cholesterol added into them, you will find that most of the phospholipids or the sphingolipids which have saturated fatty acid will tend to bind more to the cholesterol. So you can see how immediately with the arrival of the cholesterol in the membrane, all of these, okay, interact more preferentially with the cholesterol, so they all club into one domain, you see. So therefore this phase is getting separated from this phase, so this becomes one domain, this becomes another domain basically, or there is no domain over here, this is a very typical domain that has been formed. So cholesterol interacts preferentially with long saturated phospho and sphingolipids. When you say saturated, it means that the fatty acyl chains do not have cis double bonds, and therefore those are preferentially able to bind to the cholesterol. So as you can see, immediately there is rearrangement of the lipids in the membrane. All those that have saturated fatty acyl chains will go and interact specifically with the preferentially with the cholesterol. So therefore, this becomes more gel-like, this becomes more solid because it is more compact, and this becomes more fluid. This remains more fluid. So you can see how the addition of the cholesterol immediately leads to a high gel to liquid crystalline phase transition temperature, and therefore you would need a higher temperature to kind of make this more liquid, because it is in a compact form. So you can very clearly see how phase transition is something that is also influenced by the cholesterol. We all know that membranes being semi-liquid, they are able to change depending on the temperature. So if the membrane is shifted to a lower temperature, then it can become compact, or it can become it can kind of become frozen, while if it is going to a higher temperature, it can become more fluid. But if the cholesterol is present, then that transition is slower. Okay, so therefore a cell will not undergo stress, that when you expose a cell to higher temperatures, suddenly it will not get fluid because of the presence of cholesterol. So the cholesterol is actually maintaining fluidity when it is in a lower temperature, and it is increasing rigidity when it is going to a higher temperature. So it helps in either way. So it is helping in phase transitions basically. So, or rather what we can say, it is influencing or uh, or contributing to separation of phases, and that is something that is very important. Within membranes itself, you can see in certain regions where there are less amount of cholesterol, this is a different phase, this is more liquid. But when you have more number of what do you say, cholesterol present, the cholesterol would kind of bring in all the saturated fatty acyl containing sphingolipids and phospholipids together. So this is one kind of phase. So this is considered to be cholesterol poor liquid disordered membrane domain, and this is considered to be cholesterol rich liquid ordered membrane. There is more order over here and less order over here. That is something that we also saw previously. And these ordered membrane domains are also what are called as lipid rafts. These lipid rafts are small They are very micro domains, and they can be transient, present only for a certain period of time. So they contribute also to membrane asymmetry and fluidity. So this is very important. That is, if you have these microdomains being formed, obviously this being much ordered, you will find the fluidity much less over here. Okay, and also you would find that the symmetry of the membrane is no longer there. If you have a line of symmetry here, this phase is very different from this. The thickness is different over here. The thickness is different over here, and that is definitely something that is contributed by the cholesterol. Also looking at the fact that lipid rafts can be formed specifically also to by attaching to membrane proteins, integral membrane proteins, that is. And what has been observed is that cholesterol is able to induce a change in the conformation of the integral protein. See, when you have no cholesterol, you have the integral protein have a particular conformation, that is shape, but when it is interacting with the bilipid layer where cholesterol is present, you can immediately see there is a conformational change. So therefore, in this conformational change, the protein will have a different function, and in this conformational change, the protein can have no function or different function. That either is possible. So therefore, it is able to influence the functions of the membrane proteins as well, because they are able to induce a conformational change in the integral membrane proteins. So these, therefore, if you have something like this, then this forms a microdomain, and in that microdomain, the integral membrane would have a certain function. So therefore, that lipid raft is responsible for a particular function. Okay. Now, what can also happen is that the integral membranes itself can be redistributed in the entire membrane, depending on how or what is the affinity of the cholesterol to bind to the integral membrane protein. So say for example, you have this protein, okay, which can change conformation, number one. Secondly, that the distribution of this protein is greater when you have an ordered structure, while this would be present more in a disordered structure. So because cholesterol preferentially binds to this, it will be present within this microdomain region. So all of these can happen. So redistribution of integral membrane proteins can be aided or induced by cholesterol, and that would lead to dynamism as the functionality of the membrane and influence the functionality of the membrane. So let us therefore make the conclusions. Cholesterol is a sterol present in animal cell membrane. They are amphipathic with a ring structure that is flat and rigid and a hydrocarbon tail that is flexible. Cholesterol is able to interact with other lipid components to increase viscosity, decrease fluidity, and change the dynamism of the membrane. Cholesterol can be found on both leaflets of the membrane, engage with sphingolipids and membrane proteins to form microdomains or lipid rafts. One of the major roles of cholesterol has been in introducing phase changes in the bilipid layer. Here, by phase, we mean whether the membrane is less ordered or more ordered, whether it is more fluid or less fluid, more gel-like. Those are what we understand by phases. And the phase transition rates are influenced to a great extent based on the amount of the cholesterol present. So cholesterol are found embedded in the bilipid layer with only its enol group facing the surrounding aqueous environment. This enables it to bring multiple modulations to the biological membrane. It being very much integrated within the biological membrane makes a lot of difference. And this is also why we know that cholesterol is important for our body and that we need good cholesterol. Thank you.